So thank you, Ole, for that very generous introduction, and good morning, everybody, and thanks to the organizers. It's really an honor to be giving this talk. So I'm going to talk about the structure and the function and the inhibition of the HIV capsid, and that's become a very important topic because, as you just heard, Gilead Sciences has developed a very potent inhibitor of the capsid that blocks HIV replication and, more importantly, blocks HIV transmission. So the organizers asked me if I would give a bit of a historical perspective, so I'll talk about how we came to understand the structure and the function of the capsid. And then I'm going to talk about how Lenacapavir interacts with the capsid. And then I'm going to finish by talking about the thing that's most exciting in the lab right now, which is our attempt to reconstitute HIV reverse transcription and integration in a cell-free system. And as you'll see, that is also a capsid-dependent phenomenon. But I thought I'd start with the punchline. So this is the results of the clinical trials in Africa on women in high-risk areas in Uganda and South Africa, high risk for HIV. The clinical trials were run by Gilead. And what they're measuring is the incidence of infection per 100 person years. So if you follow 100 women for a year, then in these high-risk areas, on average, about 2.5 of them will become infected with HIV each year. And we have good drugs that can work in some contexts to prevent HIV transmission. They're based on the drug tenofovir. But in the field, they don't work well. You can see that they're only just a little bit better than doing nothing at all. And the reason for that is because it's very hard to remain compliant in these conditions, taking a drug that has stigma for a disease that you don't already have. And so this happens time and again. We find this in clinical trials. But if you look at the Lenacapavir arm of the clinical trials, the really remarkable result is that not a single woman became HIV positive, in spite of the fact that there were thousands of women enrolled in the trial. And so they stopped the trial, and everyone had the opportunity to go on to Lenacapavir. And this was then subsequently repeated in another large clinical trial in men and non-binary individuals. And so Lenacapavir really does block HIV transmission. And the reason it works so well, it's potent, but the real secret is very long-lasting. And so a single subcutaneous injection of the drug protects people for 6 months, and Gilead now has a 1-year formulation. And so that sort of enforces compliance, and that's the reason that the clinical trials were so successful, and it also gives the promise that we can actually change the trajectory of the HIV pandemic. And that continues to be a major problem. Every year, about 1.3 million new people are newly infected with HIV, and about 600,000 of them die. So Lenacapavir targets the capsid, and that's what we've worked on for many years, so that's what I'm going to talk about. So this reminds me to remind you that HIV has 2 states. It has a state that we call the immature virion, and this we think of as the machine that has the ability to assemble and leave the cell. It also has the infectious mature state that's shown here on the right, and that's the machine we think of as having the ability to enter a new cell and replicate. And it's the viral protease that drives this transition and gives it directionality. So when you look at the HIV virion, the thing that immediately stands out is the conical capsid, and that's what we're going to be talking about. This is a little bit more information on how the capsid is formed. So the structural protein that organizes the immature virion is called the GAG polyprotein, and it makes, and the CA region is the central region of GAG, and it makes important protein-protein interactions at the immature virion stage. When proteolysis occurs, it cuts CA at either end of the protein. That liberates a new free protein, and it reassembles to make the conical capsid. And so I'll try to use the nomenclature that I call the protein CA, and I call the conical capsid the capsid, and the capsid plus its contents, which include the viral RNA and its enzymes, the core particle. So we're fortunate to be just down the hall from, I think, the world's leading molecular animator for science, Janet Owasa, and she made this molecular animation of how we currently think about how the capsid is formed. So this is an infected cell below. These are GAG proteins at the limiting membrane of the cell. These are envelope spike proteins. There are a few longer GAG-Pol proteins. And when I run the video, what you'll see is that the GAG protein has the ability to capture the viral RNA, that's shown in blue, to bend the membrane around itself to make the immature particle, but it doesn't have the ability to pinch off from the membrane. That's driven by a host pathway called the escort pathway, as you heard from Ole. When the escort pathway works, it severs the neck of the budding virus, and now the viral immature virions are released from the infected cell. So at this stage, the viral protease, which is part of the GAG-Pol protein, and it's shown here in pink, cuts itself out of GAG-Pol, and then it cuts GAG into a series of smaller pieces. The NC piece condenses together with a viral RNA at the center of the virus, and the CA piece, as I told you, assembles into the conical capsid. And it's the capsid that is the target of Lenacapavir and what I'm going to talk about in the talk. Sorry. So when we started our work, this was a collaboration with Mike Summers' lab and a full partnership with Chris Hill's lab. We thought it was important to understand the building block of the capsid, the CA protein. We used a combination of NMR and biochemistry and X-ray crystallography to show that the protein had the shape of something like the head of a rabbit. So we could show in solution it would dimerize weakly, and the C-terminal domains, which are globular domains, form the dimer, the head of the rabbit, and the N-terminal domains, which are quite elongated, form the ears of the rabbit. So to give you some sense of scale, about 750 copies of the CA protein have to come together to make the conical capsid. And so the next question was, how does the protein go from a dimer to higher order structures? And we were very fortunate to have a talented graduate student in the lab, Sam Lee, who built on earlier work in Carol Carter's lab. And what Sam was able to do was make pure recombinant CA protein. And he found that if he raised the salt concentration and the protein concentration, then it would spontaneously polymerize into these long helical tubes. This is a cryo-EM image of the tubes. And he and John Finch together determined the structure of those tubes. They have helical symmetry, and the repeating subunit is shown here in gold, and it's a hexamer of the CA protein. And it's embedded within what we would call a P6 lattice or a honeycomb lattice, so it has 6 nearest neighbors. So there's a 6-membered ring and then 6 nearest neighbors. And what that taught us is that the protein can not only form a dimer, but it can also form a hexamer. So at about the same time, Barbie Gansler-Pornelis, another very talented graduate student in the lab, was also looking at CA proteins recombinant assembly, and she hit on the conditions under which the protein would spontaneously form a cone. And so that was an exciting day in the lab. And in those days, we used to collect our data all on film. So my contribution was to put my thumbprint on the best image she ever got of cones. And when we looked at her structures in detail, in those days the assemblies were imperfect, and sometimes we'd get tubes like Sam saw, sometimes we'd get small cones, but sometimes we'd get cones that looked essentially identical to the cone that's in the authentic virus. So this is a virus that we thin sectioned. You can see it's surrounded by the membrane, and you can see it has a dark head because it's filled with viral RNA. But they're on the same scale, and you can see that they're clearly the same structure. And so that forced us to start thinking about how can a single protein spontaneously assemble into a cone. And it was also clear that the cone was closed at both ends. So a cone has a wide end and a narrow end, and you can see that they're both closed. And so I'm going to tell you a thought experiment that explains the concept behind the model, which is a fullerene cone, and then I'll show you the model. So the thought experiment is that if you take a piece of filter paper and scissors and you cut from the edge to the center of the filter paper, you make a second cut and in the same way, and you throw away the intervening piece of pie. If you close up the two edges, then you'll get a cone. And if you do that on a featureless piece of filter paper, then you can make the first cut and the second cut anywhere you want, and so you can make a cone of any angle that you want. But if you do that on a hexagonal lattice, like the lattice that Sam saw, then if you make the first cut along a lattice line, you have to make one of the remaining cuts along one of the remaining 5 lattice lines. And so that means that you can only make cones of discrete angles, 5 discrete angles. What might be less obvious is if I make this cut and throw away that piece, I've converted this central hexamer into a pentamer because I removed one of the subunits. And this process of introducing pentamers into a hexagonal lattice introduces a property called declination, and that's the property of enclosing space. And it turns out from Euler's theorem that we know that any hexagonal lattice can be only closed if you introduce 12 pentagonal defects. And then, so that turns out to be universally true, and we sort of know it intuitively because if you look at a traditional soccer ball, we can think of it as a hexagonal lattice of white panels, and then, as you'll see if you go home and count it in your closet, it has 12 pentagonal defects that allow it to form a spherical object. So the prediction from this concept was that if we were really making fullerene cones, then there should only be 5 allowed cone angles. And Barbie was actually making cones, so she could measure their cone angles. She made a histogram. On the right, you can see the arrows are the predicted cone angles, and you can see this histogram had the predicted periodicity. And so that implied that the structure must be a fullerene cone, and by extension, that the HIV capsid would be a fullerene cone. And so this is the structure of a fullerene cone, and you can see that in the body, it's composed of 6-membered rings. And then in a conical lattice, and on the wide end, there are a total of 7 pentamers that allow to close the wide end, and 5 pentamers that allow you to close the narrow end. So the next major advance was really driven by Owen Pornelis and others, Mark Yeager and others, who were able to make the assemblies, design disulfide crosslinks to stabilize the hexamer and the pentamer, disassemble the objects, and then purify the hexamers and the pentamers and crystallize them. And that was really an important advance. First of all, it was good for us to see there really were hexamers and pentamers, but it also showed that exactly how the subunits interacted with one another. And it was also important because this allowed structure-based drug design. So Gilead Sciences could then take the hexamer, bind weak inhibitors to it, look at how they bound, and do medicinal chemistry to make them better. And so these were important advances in several ways. And they also allowed us to build now a molecular structure, an atomic level structure of the HIV capsid. And that's what you're looking at here. And as I said, the body is this hexagonal honeycomb, and the wide end closes through the introduction of 7 pentagonal defects. So it turns out to be important to understand how the hexamers pack together in the body here, and that's what I'm going to show you on the next slide. So this is now a piece of the hexagonal lattice, and you can see there are 2 hexamers next to each other shown in color. And what I'm going to take you through is the interactions that hold the hexamers together. So the first interaction, is between these 2 hexamers across a local twofold symmetry axis. And if we extract that, you can see it's made by the C-terminal domains of the CA protein. And if we then turn it on its side, you can see that that's the head of the rabbit. And so what happens is you first dimerize, that occurs through the C-terminal domain, and then the N-terminal domains are free to make hexagonal or hexameric rings, and that's what happens. Several labs, including Peijun Zhang and Angela Gronenborn and Chris Aiken, have emphasized the importance of another interaction that's made by the C-terminal domain, which is across a local threefold, and that interaction is also important for holding the lattice together. And then the real surprise from Barbie Genzer-Pornelis' work was that there's a third kind of interaction, and that's between the C-terminal domain of one subunit with the nearest neighbor N-terminal domain in the same hexamer. And so the idea is that the N-terminal domains form a hexameric ring, and the C-terminal domains form a belt around it that stabilizes that. And the reason that's important is because it creates this cleft. And the virus uses that cleft to bind host proteins that help to get it into the nucleus. And one of those proteins is called CPSF6, and you can see it fits snugly in that pocket. And the pocket is also important because that's the binding site for Lenacapavir. So just to finish this part of the talk, HIV has been a test bed for new technologies, and one of those was cryo-EM tomography. This is a beautiful paper from John Briggs and Hans-Georg Kreuzlich's lab, where they've done tomography on actual viral particles. You can see 4 different particles here. You can perhaps make out the cone. it's a three-dimensional structure, so we can slab through the virus, and you can perhaps see there was a cone as we went through. And they have sufficient resolution by subtomogram averaging to actually find the hexamers and the pentamers. And so now what you can see is that in authentic virus, the capsid structure is in fact a fullerene cone. This is sort of a classic one with 7 pentamers and 5 pentamers. But you can see that the pentamers can actually move out in different positions into the hexagonal lattice. So you can get oblong kinds of structures, and you can also get symmetrical structures where there are 6 pentamers on either end and a helically symmetry body, that just like the structures that Sam saw. So at about the same time, another postdoc in the lab, Ute von Schwedler, was doing experiments that turned out, I think, to be more important than we thought at the time. So we knew the structure of the monomer, and what she did was to design alanine scanning mutations, a total of 50 of them, that were designed to cover the surface with mutations but not disrupt the fold. She built those back into viruses, and the surprise was that 46 of her 50 mutants were down at least twofold in infectivity. And more than half of them were down more than 20-fold in infectivity. And what that told us is the virus really cares about every surface of the CA protein. And that implied that it might actually be a good drug target. And this paper caught the eye of Thomas Seelar, who's the senior vice president for virology at Gilead. He visited our lab, got quite excited about CA as a drug target, and he deserves the real credit for going back to Gilead and creating and then directing the capsid inhibitor program that led to Lenacapavir. Ute's mutants had a number of interesting phenotypes. I'll show you where they act on the next slide. I won't take you through all of them, but this cluster is an interesting one, the turquoise mutants. We could show together with Chris Aiken's lab that they actually made the capsid more stable than the native capsid, and they were also down in infectivity. So that sort of led to this Goldilocks idea that the capsid can't be too unstable, but it also can't be too stable or it doesn't function properly. So this is the viral life cycle, and I've already told you about the second half of the life cycle. That's the animation that I showed you from Janet Awasa. Once a mature infectious viral particle is produced, it then can interact with the CD4 positive T cell through an interaction with the ONV protein. That causes fusion of the virus and the plasma membrane, and that releases the core particle into the cytoplasm. So the core particle can move along microtubules, and the really exciting discovery over the last few years in the field is that can actually move to the nuclear pore, and we think largely intact through the nuclear pore. So that comes from the work of Winnie Pothak and Hans-Georg Kreuzlich and others. And during this process, the sort of most famous steps in retrovirology are happening. The single-stranded RNA is being copied into 2 double-stranded DNA. Once the core particle is in the nucleus, the DNA then has to get out of the capsid. We call that uncoating. And that DNA is then integrated into the host chromosome with the help of the viral integrase protein. And this is the reason it's so hard to cure people of HIV, because every time the virus infects a T cell, it archives a DNA copy of itself in the chromosome. So if we look at where Ute's mutants failed, some of them failed at the process of assembly, and that's because the CA protein makes important interactions at the immature lattice stage. Some of them failed in maturation, and that's because the CA protein builds the capsid. And interestingly, those were invariably blocked in reverse transcription. So that taught us that the capsid is required for reverse transcription. Some of them we now know were blocked in docking at the nuclear pore. Some of them, the very stable mutants, were blocked at moving through the nuclear pore. So we think that there's some deformability that's required to make it through this tight squeeze. And if you look at where Lenacapavir works, it works most potently at blocking passage through the nuclear pore and immediate post-nuclear entry steps. But it also works at the assembly stage and at the capsid maturation stage, and at higher concentrations, it can even block reverse transcription. And one of the secrets to the drug is that it blocks multiple stages of viral replication, and that's because the capsid participates in so many of these different stages. So this is a slide I borrowed from Thomas Seelar, who, as I said, directed the Gilead program. And they started out screening for small molecules that inhibited formation of the tubes that I showed you at the beginning. And they worked for about 2.5 years and had inhibitors, but they weren't really going anywhere, so they stopped that. And then they did something really clever, which is they had a class of molecules that was accelerating tube formation. And so they decided to focus on those. And John Link was the director of medicinal chemistry. It was a Herculean effort. They made more than 4,000 compounds. This is a measure of the EC50. How much drug does it take to inhibit 50% replication? And you can see that this is a log scale. Their 4,000 compounds got more than 5 logs of potency, and eventually ended with the molecule that we know as Lenacapavir. And it's really a remarkable molecule. It has several stereocenters. It has 10 fluorine substituents, and it's really a beautiful piece of medicinal chemistry. So Lenacapavir has several important properties. One is it binds in the pocket that I told you about. And Gilead selected for drug resistance mutations, and they all mapped immediately to amino acids that bind in the pocket, and they weaken Lenacapavir binding. But fortunately, those viruses are poorly infectious, and the reason for that is that, as I told you, the pocket is also used by the virus to bind to host factors that help it to get into the nucleus. And the consequence of using these host factors is that the binding site is very conserved across all clades and different isolates of HIV, and that's an important property for a broad drug. But what's really amazing about the drug is its potency and its longevity. And so this is a measure of potency. GSCA1 is a Lenacapavir analog. This is EC50 again, and you can see that it's about a log better than all the compounds that were being used in the clinic. But the real excitement is its longevity. So this is from the phase 1 clinical trials. Individuals got an injection at day 0, and then this is following the drug levels in the blood as a function of time. And so it goes up and then decays, but the decay is really very slow. You can see that this is a week's time scale. And so the drug was approved for use first in individuals who are infected with HIV, and now for use in prevention of HIV transmission on a 6-month regimen. And as I said, Gilead has a 1-year formulation that's just being tested now. And so this really does have the potential to change the trajectory of the HIV pandemic. So this is where we currently stand. This is a paper from Owen Pornellis' lab. We're on it too, but it's really driven by him. This is a cryo-EM image of the conical capsid. You can see it's nicely rounded. But if you add Lenacapavir, what happens is Lenacapavir binds and it flattens the lattice. And we think that's because it enforces a preferred bite angle on adjacent hexamers. And the consequence of that is it introduces lattice strains. So you get flat lattices where it binds, but then you get fissures and you get loss of the areas of great curvature, the declination sites. And so it actually acts allosterically to induce strain onto the capsid lattice and crack the capsid. And these structures are at quite high resolution with authentic viral particles, so cryo-EMs allowed them to actually visualize exactly how the drug binds. And so this is where we currently stand as of last month. The drug has been approved for prevention uses in 15 countries, including the US and also sub-Saharan Africa, where the problem is greatest. And there are 9 pending approvals. So this is about to be and being rolled out sort of worldwide. So I feel really privileged to have been part of this process from basic science to a drug that's now being rolled out worldwide. And a couple of observations. One is that the Lenacapavir is really an exceptional drug in several ways. I haven't done it justice, but it binds very tightly. It's a picomolar inhibitor. It has great pharmacokinetics, so those fluorine substituents block liver oxidation. And perhaps most importantly, it's poorly soluble. So when you get a subcutaneous injection, it sets up its own depot and then slowly dissolves over 6 months, and that's how it can last so long. I think we also now know that viral capsids can be good drug targets. I'm not sure we believed it, but I can tell you a lot of other people didn't believe that when we started our work. But I think the reason for that is that, as you saw, the capsid participates in a large number of different steps in the viral life cycle. It's really what organizes the virus. But it's also dynamic, and so it has to assemble and disassemble, and those are both highly cooperative events. And what that means is that there's only a narrow energy range over which the assembly and disassembly can take place. And so anything that perturbs the energy of the capsid can be detrimental. And a lattice is inherently an allosteric object. So if you bind in one site, you can induce strain in another. And then in policy terms, I've talked about the academic and industrial contributions, but I want to emphasize how remarkable and broad the contributions have been. So clinical care deliverers, physicians, the regulatory groups, and also community groups. They were really responsible for helping to design the trials that end up with approval in adolescent girls and in pregnant women. And then I think we sort of stand poised on a great opportunity. The drug is being shipped out by the millions. But as you may know, there are also headwinds. So USAID has been dismantled, and PEPFAR, who's the major supplier of anti-HIV drugs in the developing world, is somewhat crippled, although to their credit, they bought 2 million annual doses in September and are now distributing them worldwide. And I think it's really time for us to have resolve, because we really have the potential with this new tool to make a difference in the pandemic. So I'm just going to finish by telling you about what's exciting in the lab right now. We're trying to reconstitute and have succeeded in reconstituting the first half of the viral life cycle. So the reason for doing that is because normally a single virus infects a single cell, and that the transformations that we're perhaps most interested in happen deep inside the cell. And so we're convinced if we can reconstitute it in bulk, that we'll be able to do mechanistic studies that just aren't possible when you're looking at single infections. And we, in this case, is Devin Christensen, a research associate in the lab, and Miguel Pereira, who's a postdoc who joined him. I've already told you about the capsid, so the good news is I'm not going to tell you any more about the capsid, but I am going to tell you about the properties of the hexamer, which were discovered by other labs and are quite remarkable. So the hexamer comes together, and there are basic residues that make 2 rings. And those basic residues would repel each other, except that they create the binding sites for the small molecule inositol hexacus phosphate, IP6. And 2 molecules of IP6 bind, and that stabilizes the hexamer, and it stabilizes the capsid. So now we can do biochemistry on the capsid. And Leo James' lab showed further that it's not just a binding site, but it's actually a pore, and it can incorporate and pass DNTPs. And so now we can think about reverse transcription actually taking place inside the conical capsid, and that's what we think happens. And then this is just to remind you that the core has the capsid shell, but it also has 2 copies of the single-stranded RNA. We think the RNA is coded by the NC protein, and it has about 100 copies of reverse transcriptase and integrase. So the virus brings in the core everything it needs to replicate. So the experiment, which keeps getting better, this is the original version. We take viral particles, we permeabilize them with digitonin and melatonin. That disrupts the membrane and releases the core. If we do that into buffers that contain IP6 and NTPs, then the cores are quite stable. You can do biochemistry. And if we have DNTPs present, then reverse transcription can take place. And as I'll show you, it's really very efficient. So you have 10,000 nucleotide single-stranded RNA. It has to be copied twice to create double-stranded DNA, and I'll show you that that happens very efficiently. And then if we add a DNA target, the system will go on to do integration. And initially, the integration reaction was not very efficient. We had to add cell lysate. Then we graduated to nuclear lysate, which made it more efficient. And now we can do it with pure recombinant proteins, as I'll show you. So we're now doing the reaction where we're purifying the core particles. We can actually do ion exchange chromatography on purified core particles. And you can see that they have the protein content that you would expect, the CA and NC in bulk, and the enzymes. And to do pure recombinant system, we have to use a protein that was discovered by others called LegF. So LegF is a host protein that the virus uses to tether itself to chromatin. And it does that because the integrase complex binds to a domain that's been named the integrase binding domain. And at the other end of LegF, there's this PWWP domain that binds to chromatin, especially chromatin that has histone H3 proteins that are trimethylated on lyse 36. And so this is how the virus tethers itself to the chromatin, but also ensures that it gets integrated into active chromatin. Alan Engelman and Peter Sharapanoff have determined a structure of the relevant complex, and so we know how it binds, and they've also identified a point mutant that knocks out the integrase binding domain interaction with the intosome. And so this is my last data slide. I'll just show you that we can now do a reaction in which we start with purified cores, DNTPs, LEG-F, and NTPs, and IP6, and we have PCR primers that allow us to measure the initiation of reverse transcription. So both RNA strands initiate, but they resolve into a single double-stranded DNA, and you can see that the efficiency is about 1 double-stranded DNA per input core. And now, for the first time, we're seeing integration in this pure recombinant system, and it depends on increasing the concentration of LEG-F. So we expected that the reaction would go even better in chromatinized DNA, and that's exactly what we see. So if we chromatinize the target template, we now get about 10% efficiency, where we get reverse transcription to complete replication and integration at about 10% efficiency. And that requires LEG-F, because the point mutant in LEG-F that doesn't bind the intosome also fails to support this reaction. So we're doing a lot of this. I'll summarize some of our findings. We find now efficient ERT under optimized conditions. It's about 80% yield. What we found is that you require a capsid of optimal stability and geometry. So we think this entire reaction is taking place inside the conical capsid. We get concerted integration, so both ends of the genome integrate into the same site and with the correct local sequence preferences. That reaction is stimulated by nuclear factors. One of those nuclear factors is LEG-F, and it's stimulated by chromatin templates, and it's also stimulated by dropping the temperature. And we think there may be a phase separation reaction going on, but we have to prove that. We get overall yield of 10%. We still think we're missing additional nuclear factors that might be involved in uncoding, and so we're working hard on that problem. And so with that, I want to thank all of the people who contributed to this. So the people who are shown in yellow are the people who contributed to the original work that I talked about. The people shown in red are the people who are working on the project now. We've been very fortunate to interact with the amazing group at Gilead, and led by Thomas Seelar and John Link and Steve Yant, and that's a very large group, as you would guess, of talented people. You're looking at the University of Utah Health Sciences Center. That's our lab. That's my house. And so every morning I get to walk up over a hill and I see the Wasatch Front, and I find that quite inspiring. And I find all of these people quite inspiring. So thanks to all of them, and thanks to you for your attention.